Hello and welcome, dear students. Today we are going to discuss about the topic glucose 6-phosphate dehydrogenase deficiency. The main objectives of today's lecture are to know the basics of glucose 6-phosphate dehydrogenase deficiency, to know the history of glucose 6-phosphate dehydrogenase deficiency, to understand the causes of glucose 6-phosphate dehydrogenase deficiency, to discuss the clinical manifestations of glucose 6-phosphate dehydrogenase mutation, to discuss the genetics of glucose 6-phosphate dehydrogenase deficiency, and to discuss the pathophysiology of glucose 6-phosphate dehydrogenase deficiency. Dear students, let's start with the introduction first. Glucose 6-phosphate dehydrogenase deficiency, or G6PD, which is the most common enzyme deficiency worldwide, is an inborn error of metabolism that predisposes to red blood cells breakdown. Most of the time, those who are affected have no symptoms. Following a specific trigger, symptoms such as yellowish skin, dark urine, shortness of breath, and feeling tired may develop. Complications can include anemia and newborn jaundice. Some people never have symptoms. It's an X-linked recessive disorder that results in defective glucose 6-phosphate dehydrogenase enzyme. Glucose 6-phosphate dehydrogenase is an enzyme which protects red blood cells which carry oxygen from the lungs to tissues throughout the body. A defect of the enzyme results in the premature breakdown of red blood cells. This destruction of red blood cells is called hemolysis. Red blood cells breakdown may be triggered by infections, certain medications, stress, or foods such as fava beans. Depending on the specific mutation, the severity of the condition may vary from person to person. Diagnosis is based on symptoms and supported by blood tests and genetic testing. Affected persons must avoid dietary triggers, notably the fava beans. This can be difficult as fava beans may be called broad beans and are used in many foods. Since glucose 6-phosphate dehydrogenase deficiency is not an allergy, food regulations in most countries do not require that fava beans be highlighted as an allergen on the label. Treatment of acute episodes may include medications for infection, stopping the offending medication, or blood transfusions. Jaundice in newborns may be treated with Billy lights, that is phototherapy. It is recommended that people be tested for glucose 6-phosphate dehydrogenase deficiency before certain medications such as primaquine are taken. About 400 million people have the condition globally. It is particularly common in certain parts of Africa, Asia, the Mediterranean, and the Middle East. Males are affected more than the females. In 2015, it is believed to have resulted in 33,000 deaths. Dear students, let's now discuss about the history of glucose 6-phosphate dehydrogenase deficiency. The modern understanding of the condition began with the analysis of patients who exhibited sensitivity to primaquine. The discovery of glucose 6-phosphate dehydrogenase deficiency relied heavily upon the testing of prisoner volunteers at the Illinois State, a type of study which today is considered unethical and cannot be performed. When some prisoners were given the drug primaquine, some developed hemolytic anemia, but others did not. In spite of these results, the US military administered the drug widely during the Korean War to prevent the relapsing infection caused by Plasmodium vivax hypnozoites. Numerous cases of hemolytic anemia were observed in US soldiers of North African and Mediterranean descent. After studying the mechanism through CR51 testing, also known as 51 CR release assay, it has been conclusively shown that the hemolytic effect of primacoin was due to an intrinsic defect of erythrocytes. Dear students, let's discuss about the signs and symptoms of glucose 6-phosphate dehydrogenase deficiency. Most individuals with G6PD deficiency are asymptomatic, and most people who develop symptoms are males due to the X-linked pattern of inheritance. But female carriers can be affected due to unfavorable lionization or skewed X inactivation, where random inactivation of an X chromosome in certain cells creates a population of glucose 6-phosphate dehydrogenase deficient red blood cells coexisting with unaffected red blood cells. A female with one affected X chromosome will show the deficiency in approximately half of her red blood cells. However, in some cases, including double X deficiency, the ratio can be much more than half, making the individual almost as sensitive as males. Red blood cell breakdown, also known as hemolysis, in glucose 6-phosphate dehydrogenase deficiency can manifest in a number of ways, including prolonged neonatal jaundice, possibly leading to kernicterus, arguably the most serious complication of glucose 6-phosphate dehydrogenase deficiency. Hemolytic crisis in response to illness, especially the infections. Certain drugs, certain foods, most notably broad beans, from which the word favism derives. Certain chemicals, diabetic ketoacidosis, hemoglobinuria, red or brown urine. Very severe crisis can cause acute kidney injury as well. Favism is a hemolytic response to the consumption of fava beans, also known as broad beans. Though all individuals with favism show glucose 6-phosphate dehydrogenase deficiency, not all individuals with glucose 6-phosphate dehydrogenase deficiency show favism. The condition is known to be more prevalent in infants and children, and glucose 6-phosphate dehydrogenase genetic variant can influence chemical sensitivity. Other than this, the specifics of the chemical relationship between favism and glucose 6-phosphate dehydrogenase are not well understood. Dear students, let's now understand the causes of glucose 6-phosphate dehydrogenase deficiency. Glucose 6-phosphate dehydrogenase deficiency results from the mutations in the G6PD gene. This gene provides instructions for making an enzyme called glucose 6-phosphate dehydrogenase enzyme. This enzyme is involved in the normal processing of carbohydrates. It also protects red blood cells from the effects of potentially harmful molecules called reactive oxygen species, or ROS, which are byproducts of normal cellular metabolism or functions. Chemical reactions involving glucose phosphate dehydrogenase produce compounds that prevent reactive oxygen species from building up to toxic levels within red blood cells. If reduction in the amount of glucose 6-phosphate dehydrogenase or alteration of structure occurs due to mutations of glucose 6-phosphate dehydrogenase gene, the enzyme loses its protective role and leads to the accumulation of reactive oxygen species and thus damage of red blood cells. As a result, reactive oxygen species can accumulate and damage red blood cells. Factors such as infections, certain drugs, or ingesting fava beans can increase the levels of reactive oxygen species, causing red blood cells to be destroyed faster than the body can replace them. A reduction in the number of red blood cells causes the signs and symptoms of hemolytic anemia. Researchers believe that people who have a glucose 6-phosphate dehydrogenase mutation may be partially protected against malaria, an infectious disease carried by certain type of mosquitoes and caused by Plasmodium. A reduction in the amount of functional glucose 6-phosphate dehydrogenase appears to make it more difficult for this parasite to invade red blood cells. Glucose 6-phosphate dehydrogenase deficiency occurs most frequently in areas of the world where malaria is common. Dear students, let's understand the clinical manifestation of glucose 6-phosphate dehydrogenase mutation. Fortunately, most G6PD deficient individuals are asymptomatic throughout their life and unaware of their stats. The illness generally manifests as acute hemolysis, which usually arises when red blood cells undergo oxidative stress triggered by agents such as drugs, infection, or the ingestion of fava beans. Glucose 6-phosphate dehydrogenase deficiency does not seem to affect life expectancy, quality of life, or the activity of affected individuals. Glucose 6-phosphate dehydrogenase deficiency usually presents as drug-induced or infection-induced acute hemolytic anemia, favism, neonatal jaundice, or chronic non-spherocytic hemolytic anemia. Several clinical disorders such as diabetes and myocardial infarction or strenuous physical exercise have been reported to precipitate hemolysis in glucose 6-phosphate dehydrogenase deficient individuals. However, coexisting infection or oxidant drug exposure can be the underlying cause in these instances. The precise mechanism by which increased sensitivity to oxidative damage leads to hemolysis is not fully known. Furthermore, the exact sequence of events once an exogenous trigger factor is present is also unknown. Whatever the cause of the acute hemolysis in glucose 6-phosphate dehydrogenase deficiency, it is characterized clinically by fatigue, back pain, anemia, and jaundice. Increased unconjugated bilirubin, lactate dehydrogenase, and reticulocytosis are markers of the disorder. Carriers of the underlying mutation do not show any symptoms unless their red blood cells are exposed to certain triggers, which can be of four main types. Foods, fava beans is the hallmark trigger for glucose 6-phosphate dehydrogenase mutation carriers. Certain medicines, including aspirin, quinine, and other antimalarials derived from quinine, moth balls, that is naphthalene, stress from a bacterial or viral infection. Dear students, let's discuss the impact of drugs in glucose 6-phosphate dehydrogenase deficiency. Many substances are potentially harmful to people with G6PD deficiency. Variation in response to these substances makes individual predictions difficult. Antimalarial drugs that can cause acute hemolysis in people with glucose 6-phosphate dehydrogenase deficiency include primaquine, pemaquine, chloroquine, and hydroxychloroquine. There is evidence that other antimalarials may also exacerbate glucose 6-phosphate dehydrogenase deficiency, but only at higher doses. Sulfonamides such as sulfonamide, sulfomethoxazole, and mefenide, thiazole sulfonate, methylene blue, and naphthalene should also be avoided by people with glucose 6-phosphate dehydrogenase deficiency condition, as they antagonize folate synthesis, as should certain analgesics such as phenazopyridine and acetonidine, and a few non-sulfur antibiotics like nalidixic acid, nitrofurantoin, isoniazid, and dapsone. Resburicase, a medication that helps to clear uric acid from the blood, is also contraindicated in glucose 6-phosphate dehydrogenase deficiency. High-dose intravenous vitamin C has also been known to cause hemolysis in glucose 6-phosphate dehydrogenase deficiency carriers. Therefore, G6PD deficiency testing is routine before infusion of doses of 25 grams or more. Dear students, let's discuss about the genetics of glucose 6-phosphate dehydrogenase deficiency. Two variants, that is glucose 6-phosphate dehydrogenase A and glucose 6-phosphate dehydrogenase Mediterranean are the most common in human populations. G6PD-A has an occurrence of 10% of Africans and African Americans, while G6PD Mediterranean is prevalent in the Middle East population. The known distribution of the mutated allele is largely limited to a people of Mediterranean origins. Both variants are believed to stem from a strongly protective effect against Plasmodium falciparum and Plasmodium vivax malarial parasite. It is particularly frequent in the Kurdish Jewish population, wherein approximately 1 in 2 males have the condition and the same rate of females are carriers. It is also common in African American, Saudi Arabian, Sardinian males, some African populations, and Asian groups. All mutations that cause glucose 6-phosphate dehydrogenase deficiency are found on the long arm of the X chromosome on band XQ28. The glucose 6-phosphate dehydrogenase gene spans some 18.5 kilobase pairs long. Dear students, let's understand the pathophysiology of glucose 6-phosphate dehydrogenase deficiency. Glucose 6-phosphate dehydrogenase is an enzyme in the pentose phosphate pathway. Glucose 6-phosphate dehydrogenase converts glucose 6-phosphate into 6-phosphoglucolactone and is the rate-limiting enzyme of this metabolic pathway that supplies reducing energy to cells by maintaining the levels of the reduced form of the coenzyme nicotinamide adenine dinucleotide phosphate, or NADPH. The NADPH in turn maintains the supply of reduced glutathione in the cells that is used to mop up free radicals that cause oxidative damage. The G6PD/NADPH pathway is the only source of reduced glutathione in red blood cells or erythrocytes. The role of red blood cells as oxygen carriers puts them at substantial risk of damage from oxidizing free radicals, except for the protective effect of G6PD NADPH glutathione. People with G6PD deficiency are therefore at risk of hemolytic anemia in state of oxidative stress. Oxidative stress can result from infection and from chemical exposure to medication and certain foods. Broad beans, for example, fava beans, contain high levels of vicine, divicine, convicine, and isouracil, all of which create oxidants. When all remaining reduced glutathione is consumed, enzymes and other proteins, including hemoglobin, are subsequently damaged by the oxidants, leading to the cross-bonding and protein deposition in the red cells membranes. Damaged red cells are phagocytosed and subsequently sequestered, taken out of circulation in the spleen. The hemoglobin is metabolized to bilirubin, causing jaundice at high concentrations. The red cells rarely disintegrate in the circulation, so hemoglobin is rarely excreted directly by the kidney. But this can occur in severe cases, causing acute kidney injury. Deficiency of glucose 6-phosphate dehydrogenase in the alternative pathway causes the build-up of glucose, and thus there is an increase of advanced glycation end products. The deficiency also reduces the amount of NADPH which is required for the formation of nitric oxide. The high prevalence of diabetes mellitus type 2 and hypertension in Afro-Caribbeans in the West could be directly related to the incidence of glucose 6-phosphate dehydrogenase deficiency in those populations. Although female carriers can have a mild form of glucose 6-phosphate dehydrogenase deficiency dependent on the degree of inactivation of the unaffected X chromosome. In homozygous females, there is coincidence of a rare immune disorder termed chronic granulomatous disease, or CGD. Dear students, let's talk about the diagnosis of glucose 6 phosphate dehydrogenase deficiency. The diagnosis is generally suspected when patients from certain ethnic groups develop anemia, jaundice, and symptoms of hemolysis, especially when there is a positive family history in that population. Generally, tests will include complete blood count and reticulocyte count in active glucose 6 phosphate dehydrogenase deficiency. Heinz bodies can be seen in red blood cells on a blood film. Liver enzyme analysis can also be done. Lactate dehydrogenase, that is elevated in hemolysis, and a marker of hemolytic severity is also checked. Hepatoglobin, that is decreased in hemolysis, a direct antiglobulin test, that's Coombs test. This should be negative as hemolysis in glucose 6 phosphate dehydrogenase is not immune mediated. When there are sufficient grounds to suspect glucose 6 phosphate dehydrogenase deficiency, a direct test for G6PD is the Butler fluorescent spot test, which has largely replaced an older test, the Motulsky dye decoloration test. Other possibilities are direct DNA testing or sequencing of the glucose 6 phosphate dehydrogenase gene. The Butler fluorescent spot test is a rapid and inexpensive test that visually identifies NADPH produced by glucose 6 phosphate dehydrogenase under ultraviolet light. When the blood spot does not show fluorescence, the test is positive. It can be falsely negative in patients who are actively hemolyzing. It can therefore only be done 2 to 3 weeks after a hemolytic episode. When a macrophage in the spleen identifies a RBC with a Heinz body, it removes the precipitate and a small piece of the membrane, leading to characteristic white cells. However, if a large number of Heinz bodies are produced, as in the case of glucose 6 phosphate dehydrogenase deficiency, some Heinz bodies will nonetheless be visible when viewing RBC that have been stained with crystal violet. This easy and inexpensive test can lead to an initial presumption of glucose 6 phosphate dehydrogenase deficiency, which can be confirmed with the other tests. Testing during and for many weeks after a hemolytic episode will lead to false negative results, as the glucose 6 phosphate dehydrogenase deficient RBC will have been excreted and the young RBC, that is reticulocyte, will not yet be glucose 6 phosphate dehydrogenase deficient. False negative results will also be likely following any blood transfusions. For this reason, many hospitals wait for 3 months after a hemolytic episode before testing for glucose 6 phosphate dehydrogenase deficiency test. Females should have their glucose 6 phosphate dehydrogenase activity measured by quantitative assays to avoid being misclassified by screening tests. Dear students, let's discuss about the differential diagnosis. There are several disorders that are characterized by anemia due to the premature destruction of red blood cells. These disorders include acquired autoimmune hemolytic anemia, pyruvate kinase deficiency, cold antibody hemolytic anemia, warm antibody hemolytic anemia, hereditary spherocytosis, and sickle cell anemia. Symptoms of these disorders can be similar to those of glucose 6 phosphate dehydrogenase deficiency. Comparisons may be useful for a differential diagnosis. 6-phosphogluconate dehydrogenase deficiency has similar symptoms and is often mistaken for glucose 6 phosphate dehydrogenase deficiency, as the affected enzyme is within the same pathway. However, these diseases are not linked and can be found within the same person. Dear students, let's now discuss about the treatment of glucose 6 phosphate dehydrogenase deficiency. The most important measure is prevention, avoidance of the drugs and foods that cause hemolysis. Vaccination against some common pathogens like hepatitis A and hepatitis B may prevent infection induced attacks. In the acute phase of hemolysis, blood transfusion might be necessary or even dialysis in acute kidney failures. Blood transfusion is an important symptomatic measure as the transfused red cells are generally not glucose 6-phosphate dehydrogenase deficient and will live a normal lifespan in the recipient's circulation. Those affected should avoid drugs such as aspirin. Some patients may benefit from removal of the spleen, that is splenectomy, as this is an important site of red cell destruction. Folic acid should be used in any disorder featuring a high red cell turnover. Although vitamin E and selenium have antioxidant properties, their use does not decrease the severity of glucose 6-phosphate dehydrogenase deficiency. Dear students, let's now understand about the prognosis of glucose 6-phosphate dehydrogenase deficiency. Glucose 6-phosphate dehydrogenase deficient individuals do not appear to acquire any illness more frequently than other people and may have less risk than other people for acquiring ischemic heart disease and cerebrovascular disease. However, a recent study revealed that glucose 6-phosphate dehydrogenase deficiency increases the cardiovascular risk up to 70%. The risk conferred by glucose 6-phosphate dehydrogenase deficiency is moderate compared with the impact of primary cardiovascular risk factors. Dear students, let's discuss about the epidemiology of glucose 6-phosphate dehydrogenase deficiency. Glucose 6-phosphate dehydrogenase deficiency is the second most common human enzyme defect after ALDH2 deficiency, being present in more than 400 million people worldwide. G6PD deficiency resulted in 4,100 deaths in 2013 and 3,400 deaths in 1990. African, Middle Eastern, and South Asian people are affected the most, including those who have these ancestries. A side effect of this disease is that it confers protection against malaria, in particular the form of malaria caused by Plasmodium falciparum, the most deadly form of malaria. A similar relationship exists between malaria and sickle cell disease. One theory to explain this is that cells infected with the Plasmodium parasite are cleared more rapidly by the spleen. This phenomena might give glucose 6-phosphate dehydrogenase deficiency carriers an evolutionary advantage by increasing their fitness in malarial endemic environments. In vitro studies have shown that the Plasmodium falciparum is very sensitive to oxidative damage. This is the basis for another theory, that is the genetic defect confers resistance due to the fact that the glucose 6-phosphate dehydrogenase deficient host has a higher level of oxidative agents that are generally tolerable by the host but are deadly to the parasite. Dear students, let's now conclude the lecture. Glucose 6-phosphate dehydrogenase deficiency is a genetic disorder that affects red blood cells or RBCs, which carry oxygen from the lungs to tissues throughout the body. In affected individuals, a defect in an enzyme called glucose 6-phosphate dehydrogenase causes red blood cells to break down prematurely. This destruction of red blood cells is called hemolysis. The most common medical problem associated with glucose 6-phosphate dehydrogenase deficiency is hemolytic anemia, which occurs when red blood cells are destroyed faster than the body can replace them. This type of anemia leads to paleness, yellowing of the skin, and whites of the eyes, that is the joints, dark urine, fatigue, shortness of breath, and a rapid heart rate. In people with glucose 6 phosphate dehydrogenase deficiency, hemolytic anemia is most often triggered by bacterial or viral infections or by certain drugs such as some antibiotics and medications used to treat malaria. Glucose 6 phosphate dehydrogenase deficiency is also a significant cause of mild to severe jaundice in newborns. Many people with this disorder, however, never experience any signs or symptoms and are unaware that they have the condition. Dear students, it was all about today's lecture regarding glucose 6 phosphate dehydrogenase deficiency. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.